Quick note, a lot of this information is dated from back in 2010 when I completed my undergrad thesis, so bear with it. It's not the most recent information, but some of it still holds true. It's interesting to hear a year's work of my life condensed into 20 minutes, but here it is. Enjoy.
Schizophrenia is such a significant condition. It affects, about 1% of people globally? It's so complex, and we still don't fully grasp all the causes.
That complexity is definitely the key word. It's not, one single cause. It's almost certainly a mix of things, genetics, environment, and probably how they interact.
And that's exactly what we're digging into today, focusing specifically on the genetic and epigenetic sides of this puzzle. We've got two main sources here. One's a literature review looking at a bunch of individual studies on genes and epigenetics and schizophrenia. The other is an analysis from a really big database, the National Comorbidity Survey Replication, or NCSR.
Yeah, and they give us two really different perspectives, don't they? You've got this sort of micro-level hunt for specific genes or changes.
Which can be frustrating sometimes based on the review.
Exactly. And then you have the macro view from the NCSR looking at the whole population families, where the risk signal is, pretty hard to ignore.
Right. So our mission here is to pull out the key insights, maybe some surprising facts, from these sources. We want to understand what they tell us about the role of genetics and epigenetics in schizophrenia susceptibility. Okay, let's unpack this. Let's start with that literature review. It covers a lot of ground, looking at specific studies, and it reminds us schizophrenia isn't just delusions and hallucinations, the sort of positive symptoms.
Right. There are also the negative symptoms, which can be just as debilitating. Things like apathy, not finding pleasure in things, social withdrawal, and cognitive issues too. Thinking problems.
Yeah, and the review makes a really crucial point. Early on, current meds are generally better for the positive symptoms.
Much better. The negative and cognitive ones, less so.
Which tells you right away why understanding the biology, maybe through genetics, is just so vital.
Yeah.
If we knew the root causes better.
Maybe we could find treatments that help with everything, not just part of the picture.
Exactly. And the review also brings in epigenetics. Can you give us a quick, sort of simple explanation of that?
Sure. Think of your DNA sequence as the hardware, the basic code. Epigenetics is more like the software. It tells the hardware which genes to read, when, and how much.
So it changes how genes work without changing the actual DNA letters.
Precisely. It involves things like adding chemical tags to DNA or the proteins it's wrapped around. These changes can turn genes on or off or dial their activity up or down. And importantly, they can sometimes be inherited, but are also potentially reversible.
Okay, interesting. And the sources suggest this might be really relevant for schizophrenia.
Yeah, there's speculation it could play a big role in susceptibility. It adds another layer of complexity beyond just the raw DNA sequence.
So the review dives into specific studies. Let's walk through some. What about early attempts to find links with specific candidate genes? I see mentions of dysbindin and cytokines from 2008.
Right, those were early targets. One study looked at dysbindin in German patients, about 222 of them. Another looked at cytokines like interleukin 2 and 4 in over 500 Japanese patients and controls.
And what came out of those big breakthroughs?
Well, no, actually. Both studies, despite having decent sample sizes for back then, found no significant association between those specific genes and schizophrenia in those groups.
Huh. Okay. So a bit of a dead end there, at least in those studies.
It suggested finding single genes with strong effects wasn't going to be simple, yeah. The weaknesses noted were things like, for the German study, maybe the narrow ethnicity reduced variation, and for the Japanese one, they didn't use standardized interviews, which could affect results.
All right, so if single genes were tricky, what about bigger genetic changes like copy number variants, CNVs, where bits of DNA are deleted or duplicated?
Good question. A big study in 2009, Rajesco et al. Looked into that. They examined genes called Nerexins NXRN1, 2, and 3, which are important for brain cell connections, synapses.
And they use a huge sample, right, across Europe?
Massive. Nearly 3,000 patients and over 33,000 controls from 7 populations. Very large scale.
So did that yield anything?
It did. They found quite a few deletions and a smaller number of duplications specifically in the NXRN1 gene among the patients. They didn't find CNVs on the other 2 Nerexin genes, though.
Okay, so a potential link there with NXRN1 involved in brain wiring.
Exactly. It was preliminary evidence. But what's fascinating here is, even with that huge sample, the study noted they couldn't really pin down the exact level of risk these CNVs conferred or how common they were overall in causing the disease. Still some uncertainty.
Interesting. So a hit, but maybe not a home run yet. What about looking even broader? Genome-wide association studies, GWAS, looking for those small variations, SNPs, across the whole genome.
Yep, another 2009 study, Need et al., did just that. They looked genome-wide for both SNPs and CNVs in large cohorts from Germany, Central Europe, Scotland, Northern Europe.
A wide net, and what did they pull in?
Well, the main takeaway was finding no highly significant genome-wide associations for SNPs at that time.
None, even with a large sample.
They concluded their sample, while large, probably still wasn't powerful enough to reliably detect risk factors that only have very small individual effects, which, is often the case with complex traits like schizophrenia, lots of genes, each adding a tiny bit of risk.
Right. So the hunt for specific genetic variants, big or small, was proving really challenging based on these studies. But you mentioned epigenetics earlier. Did studies look directly at that layer?
They did, and this is where things get quite intriguing, I think. There was a very early, very small study back in 2003 by Petronas and colleagues.
What do they do?
They looked at epigenetic differences, specifically DNA modification in the dopamine D2 receptor gene. Dopamine's a key neurotransmitter implicated in schizophrenia. And they looked in just 2 pairs of identical twins.
Identical twins, so same DNA.
Exactly. But crucially, these twins were discordant for schizophrenia, meaning one twin in each pair had it and the other didn't.
Wow. So what could they possibly find with just 4 people?
Well, remarkably, they found a significant epigenetic difference in that dopamine receptor gene between the affected twin and the unaffected twin in both pairs.
Really? That's striking.
It is. Now, obviously, you absolutely cannot generalize from N4. Huge caveat. The study itself notes tiny sample, potential confounders from individual histories, things like that. But it provided really powerful preliminary evidence, a strong hint, that epigenetic factors, things beyond the DNA sequence, might influence whether someone actually develops schizophrenia, even with the same genes.
That makes sense. A potential explanation for why one identical twin might get it and the other doesn't.
Mm-hmm.
Did other studies follow up on this epigenetic angle?
Yes, later studies looked at post-mortem brain tissue, which is obviously challenging research but can provide direct biological evidence. A 2008 study by Mill et al. Examined DNA methylation patterns.
Methylation, that's one of those epigenetic tags, right?
Exactly. They looked in the frontal cortex of people with schizophrenia, bipolar disorder, and controls. They found methylation patterns in schizophrenia were significantly correlated between males and females. And interestingly, they found a correlation between methylation in a specific gene region, MEK1, and the amount of antipsychotic medication the individuals with schizophrenia had taken during their lifetime.
So a link between an epigenetic mark and medication history.
Yeah, suggesting maybe the illness or the treatment or both could be associated with these epigenetic changes in the brain. Again, preliminary evidence, smallish sample, 35 in each group, potential confounders noted.
Okay, and there was another one looking at a specific enzyme involved in methylation.
Right. Veldick et al. In 2007, they focused on DNA methyltransferase 1, or DNMT1. It's an enzyme that adds those methyl tags to DNA. They looked at its expression levels in specific brain cells, GABAergic neurons in the prefrontal cortex.
GABA neurons are important inhibitory cells, right? Often implicated in schizophrenia theories.
Correct. And they found increased DNMT1 expression in those GABAergic neurons, specifically in the individuals with schizophrenia compared to controls. They also looked at people with bipolar disorder with psychosis, but the increase wasn't significant there.
Suggesting maybe this DNMT1 increase in those specific cells could be somewhat specific to schizophrenia.
That was the implication. It hinted that the machinery controlling epigenetic marks might be altered in schizophrenia, potentially affecting inhibitory brain circuits. But again, small samples, potential confounders like substance abuse history were limitations.
So these epigenetic studies, while often small and preliminary, seem to offer a different, potentially fruitful avenue looking at gene regulation.
It shifts the focus a bit.
Did any studies try to connect specific gene variants back to actual brain structure changes seen in schizophrenia?
Yes, a study by Zinkstock et al. In 2008 tried to do that. They looked at genes on chromosome 22q11, a region known to carry risk for schizophrenia. Specifically, they looked at COMT and PRODH genes.
COMT is involved in dopamine breakdown, right?
Yeah, and PRODH in proline metabolism, both relevant to brain function. They looked in about 50 patients with schizophrenia or schizoaffective disorder and searched for links between variations in these genes and brain anatomy differences measured by MRI.
And did they find any connections?
They reported some associations. A specific variation, an SNP in the COMT gene promoter, was linked to increased gray matter volume. And 2 different PRODH SNPs were associated with reduced white matter in the frontal lobes bilaterally.
Interesting. So specific gene variants potentially influencing brain structure in regions relevant to the disorder.
Potentially, yes, but big caveats here. Very small sample and crucially no control group in this particular study. So the findings were highly tentative, just suggesting these genes might affect brain regions linked to the phenotype.
Got it. So a lot of different angles being explored in the literature review: candidate genes, CNVs, GLBS, epigenetics, brain structure links. Were there other notable findings mentioned?
Yeah, the review touches on a few others that found preliminary associations. A study by Vaza et al. Looked at a region on chromosome 15q26 in a genetically isolated population in Italy and found a link.
Isolated population helps with genetic homogeneity.
Right, but might limit generalizability. Then Souza et al. Found associations with the GSK3 gene, particularly related to clozapine response, though their sample wasn't very representative. Liu et al. Found a link with the NOTCH4 gene region in Taiwanese families. We mentioned this one earlier. And Ma et al. Investigated a gene called PLXNA2 on chromosome 1q32, finding an association in both homogeneous and diverse samples, though the patient group came from a specific drug trial, which again raises representation questions.
Wow. Okay, so a whole range of genes and regions flagged as potentially interesting, but often with question marks. What's your big takeaway from this collection of studies in the review?
What's fascinating here is just seeing the sheer breadth of the search, Researchers were really casting a wide net, specific genes, big deletions, tiny genome-wide variations, these epigenetic marks, and the results reflect that complexity.
Yeah, a real mix.
A real mix. You have studies that just didn't find anything for their chosen target. You have hints of associations, like with NXRN1 or COMT-PRODH brain links. You have these intriguing epigenetic leads with MEK1 or DNMT1, but almost always with caveats.
And what were the most common caveats or limitations that kept cropping up across these studies, according to the source?
Sample size was a big one. Even when studies seemed large, for the type of analysis like GeoWiz or detecting small effects, they often weren't quite large enough. Another major theme was representativeness.
How so?
Many studies focused on specific ethnic groups, which is good for reducing genetic background noise, but means findings might not apply elsewhere. Or they used patient samples that weren't typical of everyone with schizophrenia, like only those in a drug trial, or those who didn't respond to certain meds.
Gotcha. And confounding factors.
Yeah, things like medication history, other illnesses, substance abuse, even cause of death in the postmortem studies. These could all potentially muddy the waters and influence the results. It's just really hard to control for everything.
Okay, so the specific gene and epigenetics hunt, as presented in this review, is clearly complex. Lots of leads, but also lots of challenges and mixed results. So, let's shift gears. What happens when we step back and look at the bigger picture, using that large population database, the NCSR?
Right. This takes us to the second source, the analysis of the National Comorbidities Survey replication. And this is a very different beast. It's designed to be a snapshot of the entire U.S. adult population, a probability sample.
Tell us a bit about who was in this NCSR sample, based on the description.
It was huge, over 20,000 people. Pretty balanced gender-wise, maybe slightly more female. Average age around 43. Good geographical spread across the U.S., Northeast, Midwest, Southwest. And diverse ethnically, too, though the source notes non-Latino whites and African Americans were large groups, and English and Spanish were common languages. Crucially, diagnoses were based on standardized DSM-IV criteria.
So a really robust representative sample.
Yeah.
What did this analysis find about just how common psychotic experiences are? Not necessarily a full diagnosis, but the experiences themselves.
This was quite striking. They found that within the past 12 months, 2% of the U.S. population reported having at least one psychotic experience.
2% in a year. Okay. What about over their lifetime?
That figure jumps significantly. 5.3% reported experiencing at least one psychotic symptom like hallucinations or delusions or disorganized thinking at some point in their life.
5.3%. That's what, 1 in 20 people?
Roughly, yeah. The source breaks it down further. Lifetime visual hallucinations were reported by 4%, auditory by 2.4%, disorganized thinking and unrealistic thinking both around 1.1%.
That's much higher than the 1% figure for diagnosed schizophrenia.
It is. And if we connect this to the bigger picture, this 5.3% lifetime prevalence of any psychotic experience suggests that these symptoms exist on a kind of spectrum in the population. The NCSR analysis itself points out that this finding supports the clinical observation that people who do get diagnosed with schizophrenia usually experience multiple symptoms or episodes over time.
Okay, that makes sense. Now here's where it gets really interesting. What did the NCSR data reveal about heritability? Did it show that schizophrenia runs in families?
It showed very clear evidence for that. They asked participants about schizophrenia and their close relatives, parents, siblings, children.
And what was the prevalence among those close relatives?
They found 1.3% of respondents reported having a close relative diagnosed with schizophrenia. When they asked about close relatives who had received treatment for schizophrenia, it was 1.4%, and relatives whose schizophrenia interfered with their lives was 1.5%. Relatives hospitalized was 1.4%.
So consistently around 1.3 to 1.5% of people in this representative U.S. sample had a close relative affected by schizophrenia.
Exactly. And remember, the prevalence in the general population is estimated around 1% or maybe even slightly less. So having it in a close relative clearly represents a significantly increased prevalence.
That's a powerful population-level signal for heritability, isn't it?
Absolutely. And the authors of the NCSR analysis explicitly state this in their conclusions drawn from their own findings. They say these results provide further evidence for heritability.
Did they connect it back to any other research?
Yes, they specifically mention that their finding is congruent with previous research, and they even cite the Liu et al. Study, the one from the literature review that found an association with the NOTCH4 gene region in Taiwanese families.
Interesting. So the micro-level study finding a potential genetic link in families gets support from this macro-level population data showing that family link is definitely real and significant in the U.S.
Precisely. There's a nice convergence there. Now, the NCSR analysis does acknowledge limitations, like the possibility of reactivity, people perhaps being hesitant to report mental illness in themselves or family members in a survey setting, social desirability bias.
Sure, that's always a potential issue with surveys.
Right. But they conclude that even with that potential underreporting, the observations, especially the higher rates among relatives, tentatively but strongly point towards the importance of genetic factors in susceptibility.
Okay, so let's try and synthesize this. We've explored the literature review, which showed researchers hunting for specific genes and epigenetic factors, finding some leads like NXRN1, MEK1, DNMT1, COMT-PRODATE links, GSK3, NOTCH4, etc., but facing challenges with mixed results and study limitations.
A complex, sometimes messy search at the molecular level.
And then we have the NCSR analysis using broad population data, which clearly showed psychotic experiences are relatively common, and more importantly, provided strong evidence that schizophrenia risk is significantly higher in close relatives, supporting heritability.
Yeah, a clear signal from the population level.
So what does this all mean? When we put these specific sources together, what's the main takeaway?
I think the big takeaway is that genetic and epigenetic factors are definitely implicated in schizophrenia risk. There's just too much evidence pointing that way, both from the specific, albeit sometimes inconsistent, findings in candidate gene or epigenetic studies.
And from that really robust heritability signal seen in the large-scale NCSR population data, the family connection is undeniable.
Exactly. But it also highlights just how hard the science is. Finding the specific biological mechanisms, the precise genes or epigenetic changes responsible, and how they work across diverse people, that's proving incredibly difficult. The studies show lots of potential candidates, but also lots of inconsistencies and limitations, especially around sample size and representation.
So the what that genetics matters seems clear from these sources. The how, the specific genes and mechanisms, is still very much being worked out.
That sums it up pretty well based on this material. This raises an important question. Think about this tension, right? The NCSR data gives us this really strong population-wide evidence for heritability. Schizophrenia clearly runs in families. The risk for close relatives is significantly elevated. It's a very clear signal.
Undeniable from that data.
But then look back at the literature review. When scientists try to zoom in and find the specific genes or the specific epigenetic switches responsible, it gets really murky. They find hints, maybe some genes pop up in one study but not another, or the effects seem small or only appear in certain populations. It's been incredibly hard to consistently nail down specific factors with big, clear effects.
Yeah, that contrast is striking. Strong general signal, but elusive specific culprits.
Exactly. So what might that disconnect tell us? Does it suggest that maybe schizophrenia isn't caused by just one or a few bad genes with strong effects? Could it be, perhaps, that the genetic risk comes from hundreds, maybe thousands, of different genes, each contributing just a tiny, tiny bit of risk?
Like a cumulative effect of many small variations.
Possibly. Or maybe it's about complex interactions between many genes. Or perhaps it underscores just how crucial those epigenetic factors are, acting alongside genetics, and how environmental influences might interact with both throughout life to ultimately determine risk. It points towards a really complex architecture.
It definitely suggests the genetic story is far from simple. Something complex is going on that makes it easy to see the family link at a distance, but hard to dissect up close.
That seems to be the challenge highlighted by putting these sources side by side.
A lot to think about there. It really emphasizes the complexity of genetic research in mental health. Thanks for joining us on this deep dive today.
Thanks for listening to this. It was interesting to upload the entirety of my thesis and have Google's AI, NotebookLM, translate it into this podcast format and listen back to it. It's funny to hear a year of your life condensed into 20 minutes. So if you like this, like, comment, subscribe, and don't forget to share it. Thank you.